It's actually quite a bit emotional because I see many colleagues who supported me in the very beginning in this NFP 50, and this was my first contact with endocrine disrupting chemicals, and our group is still working on this. I thought to present a bit different things, not talking on chemicals disrupting cortisol action, but rather on limitations on in vitro assays. So if you listen to Leo and Sarah, we identify mixtures and individual chemicals, but then we would like to understand also the mechanisms, how they cause disease. So we have chemicals that are called endocrine disrupting chemicals, and I think the second part of this sentence that I need to cause adverse health effects is very important, at least for me. And now we have rather few compounds that are convincingly shown, not just association-wise, that they cause adverse health effects, but we have, I think, hundreds of chemicals that most likely cause adverse health effects, but we have to show that and understand the underlying mechanisms. And many studies are coming from animals or in vitro studies from other species, so we need to understand also species differences. Very few studies on humans because analytics is often not developed well enough, and also hormonal systems, because we should not only focus on 6 steroid hormones. So in order to identify hazardous substances, we need in vitro assays, and also to understand the mechanisms, but the predictivity of such assays is often very low. And I would like to discuss some issues that can be responsible to the observed large lab-to-lab differences. And one issue is that the assay protocols are not sufficiently described. So if we just take cortisol as an example, we have cortisol being regulated by the hypothalamic pituitary adrenal axis. Then we have the biosynthesis in the adrenals. Cortisol is released in the bloodstream, where it's bound about 97% to cortisol binding globulin and albumin. We have transport into the target cell or efflux. For example, in the brain, we have an efflux of cortisol, but none of corticosterone. And there, corticosterone is actually important to also regulate glucocorticoid and mineralocorticoid receptors. Then we have another level, the pre-receptor control or intracrinology, where we have enzymes that convert inactive to active glucocorticoids. And finally, we have hepatic catabolism and excretion. So once the receptor is activated, we have also a feedback loop, and this regulates then the synthesis as well. So if we look at EDCs, we can potentially interfere with all these different levels. But if we now look at this system, we have one cell-based assay, this H295R adrenal assay. I come to that later. That is actually validated for testosterone and estradiol as readout, but it's mainly an adrenal model. And I hope all in the room here will not produce testosterone and estradiol in the adrenals. Then there is currently a validation of a glucocorticoid receptor transactivation going on by the OECD that should soon be available. Now, how can a certain compound interact with a given target, one of these at the many different levels? So it could affect directly by binding to the enzymal receptors. That's what we mostly look at. But it could also interfere with post-translational modification, for example, signaling, as Sarah has shown, through GPCRs, and then with protein kinases and phosphatases. Then we could affect the protein stability, intercellular distribution, protein-protein interactions, or in a sensitive period also by epigenetic modifications. So this brings us to some limitations of transactivation assays and protocols. What cell lines should we choose? what promoter-reporter gene. For example, I mentioned the GR Calyx assay. Why do we there look at the HeLa cell, or why do we look at the promoter of MMTV, or why do we overexpress the receptor but not other associated proteins? Finally, we need also to look at medium composition. Do we add serum? Do we not add serum? So some people told me, you need to add serum, otherwise it's not physiological. But the epithelial cells in my kidney hopefully never see serum. Otherwise, I might have a big problem. When I started in this field, and I started from nephrology, looking at steroids, then I saw the glucocorticoid receptor has one protein. But it's not one protein. This is this, as an example, whole complex where the receptor can form either dimers, so homodimers or heterodimers. We have many other transcription factors that are highly gene-dependent. We have coactivators, co-repressors, and that is the whole complex that is actually highly cell and target specific. So one example that I always present my students is the one of the selective estrogen receptor modulator, tamoxifen, and I ask them whether it's an agonist or an antagonist. And after listening to the oncology lecture, they always say, of course, it's an antagonist. We treat breast cancer patients that have estrogen receptor alpha positive breast cancer. But in the uterus, it's an agonist, and actually enhances the risk for endometrial cancer. Now, the question is, of course, what cell line should we choose for a transactivation assay? A breast cancer cell or endometrial cell? And how should we test the compound X that we know nothing about it? Then I would like to look at another level, intracrinology. So we have two pre-receptor enzymes that control inactive to active glucocorticoids, so I must say cortisone and also prednisone, that you maybe know from the doctor if you have an inflammation. So cortisone and prednisone are completely inactive. But cortisol, prednisolone, they're highly active to bind glucocorticoid receptor and regulate the downstream genes. Now we have these two enzymes, the 11-beta-HSD1, that if the doctor gives you prednisone, you will rapidly convert it to prednisolone in the liver, and the 11-beta-HSD2 in the kidney and the colon will inactivate cortisone to cortisone. And this avoids cortisol, that we have about 500 times more than aldosterone, to activate the mineralocorticoid receptor in these tissues. This allows aldosterone to regulate electrolyte homeostasis in the body. So if we have cells that express none of these pre-receptor enzymes, then the cell is dependent on extracellular cortisol. If a cell expresses 11-beta-HSD1, then it has its own production of cortisol from circulating cortisone, because cortisone binds about 50% to proteins in the circulation, but cortisol about 97-98%. So you have a pool of free cortisone that can be converted to the active cortisol. And cells that express 11-beta-HSD2 can inactivate cortisol, and they're only activated at stress levels or at peaks during the circadian rhythm. So we have some different cell types and situations that respond then differently to corticosteroids. For example, the kidney cell, renal cortical collecting duct cells express 11-beta-HSD2, and this renders specificity of the mineralocorticoid receptor for aldosterone. The cardiomyocyte has actually both receptors, but none of the pre-receptor enzymes. And there is evidence that in this cell type, cortisol may act as an antagonist. Then the macrophage, important in inflammation, we have the co-expression of the two receptors with 11-beta-HSD1. And here the two receptors balance a pro- and anti-inflammatory effect on these cytokines. So they have opposite effects, and the pre-receptor enzyme can then put the balance. So what cell background should we choose for MR and GR transactivation assays? I think this very much depends in which situation we want to look at, inflammation or hypertension or then cardiovascular disease. So probably most of us here in the audience have already tasted some licorice. In Basel we have the Messe, so we eat a lot of licorice. And this, for example, in pregnancy, might not be very good because this can activate then the mineralocorticoid receptor, but also glucocorticoid receptors. If you activate excessively the mineralocorticoid receptor, you get hypertension and hypokalemia. And this can be characterized in the clinics by measuring low renin and low aldosterone levels because cortisol floods the MR. Another important role of this enzyme is in the placenta. So the mother has about 10 times more cortisol than the fetus, and this is due to the placental barrier, where cortisol is inactivated to cortisone, and then the fetus is protected. If we have a disruption of this barrier, for example, by eating a lot of licorice or having some other inhibitors, then this can no longer be inactivated and the fetus is exposed to excessive amounts of cortisol that then can change, has epigenetic changes and others that lead to a higher risk of cardiometabolic disorders, also neuroendocrinology and behavioral disorders. So we have to avoid the non-intentional inhibition or also downregulation, not only the inhibition of this enzyme. Now we did a transactivation assay to look at the effect of this 11-beta-HSD2. To the very left, you see the mineralocorticoid receptor. We added 50 nanomolar cortisol. We see a potent activation in all these three cell types that are used as working horses. We can then express and co-express different enzymes. If we co-express with 11-beta-HSD2, there is a full protection by this enzyme because cortisol is inactivated to cortisone. Now when we added an 11-beta-HSD1 inhibitor, a very potent inhibitor, it's an anabolic steroid, fluoxymesterone, we see still a protection in HEC cells and COS1 cells, but a very potent activation in V79 cells. And actually the same we observed also when using an systemically used azole antifungal, posaconazole, that showed a very active or a very high activation of the mineralocorticoid receptor. So if we had used only HEC cells or COS1 cells, as we did initially, we would have concluded that fluoxymesterone and posaconazole would not inhibit the 11-beta-HSD2. However, if we use this system, V79, then we conclude that this is active. So what cell background, again, we need to choose is very important. Now here we have a comparison of the inhibition in different cell types. So the HEC cell, we see basically no inhibition of the enzyme. This is a colon cancer cell, this is a breast cancer cell. You see different sensitivities. Here we see an inhibition, and here with a weaker inhibition. Fortunately, different from the transactivation assay, we can use cell-free assays, and this is what we did with a lysate of the cells here expressing 11-beta-2 that do not respond, and we see an IC50 of 80 nanomolar. So this is a huge disadvantage, of course. If you use transactivation assay, we have the cell background. And this is most likely due to efflux transporter. As for example, in the brain, cortisol can be efflux, some substances can be efflux, but this is highly cell and tissue dependent. Now another point is the composition of medium. We were actually establishing an assay for the 11-beta-HSD1 that converts cortisone to cortisol. At that time, every second pharmaceutical company was developing inhibitors because they wanted to prevent metabolic disease and obesity. And actually, we gave this assay to a pharma company in Switzerland. They had another company in the US. And actually, we had seen in using lysates of these cells expressing this enzyme, very potent inhibition by glyceridinic acid, all the three groups. But when we use carbenoxolone, we and the Basel group had seen potent inhibition by carbenoxolone that is related and is used to treat stomach ulcers. And the US group had seen no inhibition at all. And they told us that our assay is useless. Now we wanted to compare really the details of the protocol and what we had seen. We did not add albumin to the lysate, whereas the US colleagues added BSA to the lysate. This is often done to stabilize proteins that they're not getting inactivated. And so if we then look at the serum binding, we see that glyceridinic acid, again, like cortisone, shows a very low protein binding, about 50%. But here in carbenoxolone, we have 95, or I think even more, percent binding to the serum. So basically, this explains why they had not seen any activity and we had seen activity. So the question is, or more the hint is that the medium composition must be really well defined. Now I switch to another topic. This is more male reproductive toxicology, where we can also use cell-based assays. So here, if you look at the Leydig cells, we have two key enzymes. The cytochrome P450 CYP17A1, this is essential for the production of all the steroids, all the androgens, and 17-beta-3 is producing testosterone in the terminal step. We have currently no human Leydig cell system available. As alternatives, we can use mouse Leydig cells like this, and there were over 40 scientific publications that show testosterone production and interference of compounds with testosterone production using antibody-based quantification. We then used mass spectrometry-based methods, and we detected very low or no testosterone being produced. What we had seen that all these three cell lines can nicely produce steroidogenesis down to progesterone, and this may be useful, but we have to know this limitation. When we then looked at the gene expression, we see that cytochrome P450 CYP17A1 is highly expressed in the testis, but show moderate or low expression in the cell lines. 17-beta-HSD3 is highly expressed in the testis, not at all in these three cell types. This is an enzyme that can also produce testosterone, but mainly dihydrotestosterone. It's the analogous enzyme to AKR1C3 in humans. So we need to use appropriate analytics, mass spec-derived methods and not antibody-derived methods. Then, as an alternative for male reproductive toxicity, often the OECD guideline using the adrenal cell model, H295R, is used, that is validated for testosterone and estradiol as a readout. So I mentioned that hopefully none of us in the audience produces testosterone or estradiol in the adrenals, but this cell line does. So it expresses AKR1C3, but not 17-beta-HSD3, to produce low levels of testosterone. And we have to remember that this cell line was derived from a female patient suffering from an adrenal tumor. So here again, it's the problem that tumor cells often produce steroid hormones. And again, when we looked at the expression, these cells express basically the wrong enzyme, AKR1C3, that is mainly expressed in the prostate, but do not express at all the 17-beta-HSD3. So if we see changes that are upstream of the last step, then our conclusions may be okay. But if we look at the last step of testosterone production, then we look at the wrong enzyme, and probably also the pathways that regulate these enzymes is different. So to conclude, the predictivity of our available in vitro assays is low, and this is limiting also the use of the obtained data for regulators, but we Also, we'll have difficulties in developing norms because we need to define and to characterize them. So this explains also part of the lab-to-lab differences. We need to better describe our protocols and analytical methods. And these in vitro assays so far cover only a few levels, so we need to cover also all the other levels that I showed in the beginning. And then tissue and cell-specific differences, also species differences need to be considered. And for the correct interpretation of the results, this requires a detailed understanding and a multidisciplinary approach. With this, I thank the people from my group, the funders who support us over all these years, and many colleagues. And with this, I thank you for your attention.